Metabolism encompasses all the processes and pathways needed for cell survival. These processes can be classified as catabolic processes or anabolic processes. Catabolic processes involve the breakdown of dietary components with the purpose of producing energy, most likely NADH and FADH2, which are eventually converted to ATP, a more readily usable form of energy. Anabolic processes will use the ATP produced from catabolism to synthesize cellular components like proteins, carbohydrates, lipids, membranes, nucleotides, nucleic acids, and other macromolecules, as well as maintain other cellular processes. Metabolism is compartmentalized. Some of the most important anabolic pathways occur in the cytoplasm. This includes glycogen, fatty acid, triacylglycerol, cholesterol, and nucleotide biosynthesis, as well as amino acid biosynthesis. On the other hand, some of the most important catabolic processes occur in the mitochondria, including pyruvate dehydrogenase, TCA cycle, fatty acid beta oxidation, and glutamate dehydrogenase, with glycolysis, glycogenolysis, and HMP pathway occurring in the cytoplasm. It is noteworthy that the urea cycle and gluconeogenesis start in the mitochondria and continue in the cytoplasm. Glucose has at least 4 uses in the cell. The first one is through glycolysis to produce energy and 2 molecules of pyruvate. The second one is in storage of glucose as glycogen, which occurs in both liver and muscle. The third one is in the hexose monophosphate pathway or pentose phosphate pathway, which produces NADPH and pentose phosphate or ribose phosphate needed for nucleotide biosynthesis. The fourth important use is in glycosaminoglycans, which are important extracellular matrix components, and the glycolipids, which are plasma membrane components. Fatty acids play at least 4 important roles in energy production, in energy storage, in membrane synthesis, and in inflammation. Fatty acid beta oxidation in the mitochondria is the major energy producing pathway from dietary and stored lipids. Fatty acids are stored in the form of triacylglycerol in adipocytes and provide many tissues with the energy they need during fasting. Phospholipids, the major membrane components, are made from fatty acids and glycerophosphate. The essential fatty acid, linoleic acid, is the precursor of arachidonic acid, which in turn is the precursor of all eicosanoids, which are key mediators of inflammation. There are 4 distinct uses of amino acids from dietary proteins. First one is to provide amino acids to rebuild tissue proteins that have turned over earlier. The second important purpose is to provide the carbon skeletons from amino acids to make glucose in the liver to restore blood glucose. The third important use of amino acids is in the synthesis of purines and pyrimidines needed for nucleotide synthesis. The fourth important use of amino acids is in specialized products like heme, catecholamines, and porphyrins. you like this video, please subscribe. Thank you. Cholesterol is a unique sterol and is essential for at least 4 distinct cellular functions. Cholesterol is needed for the synthesis of bile acids and bile salts, which are needed for emulsification of dietary lipids, digestion, and uptake. Cholesterol is needed for membrane synthesis and for the maintenance of membrane fluidity. Cholesterol is the precursor for the synthesis of vitamin D. And cholesterol is the precursor for the synthesis of all steroid hormones, including cortisol, androgens, and estrogens. And the reason we focus on the pancreas, because it is the source of at least 3 important hormones. And obviously, as you remember from signal transduction, most of our metabolism is dependent on the levels of various hormones in the bloodstream, whether you have high insulin or you have high glucagon. So the pancreas is the source of 3 important hormones that regulate metabolism: insulin, glucagon, and somatostatin. We'll mainly talk about insulin and glucagon in the context of their ability to change and regulate metabolism. So hence the importance of the pancreas as the source of these hormones. Another important consideration in metabolism is what's available in the bloodstream. Is there a lot of glucose? Is there a lot of amino acids? Is there a lot of tags in the form of chylomicrons? So that determines what metabolic pathways are activated and or inhibited. So that's very important consideration. Level of hormones, level of substrates, if you want, energy substrates in the bloodstream. And third, there's important input from the nervous system. The nervous system, through the hypothalamus, will direct the release of specific hormones like cortisol, like norepinephrine and epinephrine. And all of these can contribute to the overall regulation of metabolism. Okay? So input from the nervous system is very important. This is specifically important in trauma, stress, fight or flight conditions. The input from the nervous system is extremely important. So that will affect metabolism significantly, and we'll cover some of that. In the well-fed state, elevated glucose in the bloodstream will trigger the release of insulin from the pancreas. Insulin will specifically increase the uptake of glucose by muscle and adipose tissue. In the liver and muscle, insulin will activate glycogen synthesis and glycolysis while inhibiting glycogenolysis. In adipose tissue, insulin will increase triacylglycerol synthesis and inhibits lipolysis by inhibiting hormone-sensitive lipase. The brain will use the ample supply of glucose from the bloodstream, oxidizing it all the way to CO2 and producing 38 ATPs in the process. During fasting, a drop in blood glucose will trigger the release of glucagon from the pancreas. Through the hypothalamus, the brain will direct the production of cortisol, epinephrine, and norepinephrine by adrenal glands. In the liver, glucagon and epinephrine will stimulate glycogenolysis and gluconeogenesis and resupply the blood with glucose, as well as ketone bodies. The liver and muscle will switch to using fatty acids provided by lipolysis of triacylglycerol in adipose tissue. The brain can only use glucose and ketone bodies, but not fatty acids for its energy needs. If you like this video, please subscribe. Thank you.